Hello everyone, and welcome to this video podcast going over Module 1.2. In this module, what you're going to do is you're going to learn about how we extract DNA from tissues. In this particular case, we're going to learn about extracting DNA from plant tissues. But the process isn't much different if you're going to extract it from other tissues. Now, plant tissue does turn out to be a little harder to get DNA from, say, than an animal cell because of the cell wall. But the process is very similar. And then we're going to learn about PCR. We're going to learn about how PCR is set up and how it's used and what it will produce at the end of it. And then in our next lab, we'll learn how we analyze the PCR results using gel electrophoresis. And then we'll talk about how it's important in this case, in this particular case, to use something we call nested PCR. And then we'll look at sequence data for the GAPC gene from kale, cabbage, and our control, Arabidopsis, to see how closely related this gene is in these different species. The plan for Lab 1.2 is to do the lab quiz. Now, you may have already done it because it's part of the whole week 1 lab, or maybe you're breaking it up and now it's time to do the next 5 questions. However it is you're choosing to do that this semester. We're going to talk about DNA extraction, we're going to talk about PCR, and I want to remind you that due at the end of the week, Thursday, well, not really the end of the week, Thursday, at 11:59 p.m., you'll have to watch this video, and then you'll need to complete these 2 worksheets, 1.2, Part 1 and Part 2, and then there's a separate lab activity, 1.2, which is also found in the same worksheet package, so don't look anywhere else for it. And I don't know why I mentioned this twice, but watch the PCR video here. As soon as you watch it, it'll give me the indication that you watched it, and you'll get those points automatically. Okay, so we're in 1.2, as I said. We're going to talk about DNA extraction and PCR. Now, even though you're not going to be doing these actual steps, I do want to talk about them so that you're able to complete the worksheets, and also so that you understand some basic principles for when you do have an opportunity to work in a lab. Some of the research we do, we don't really always have to wear gloves, but other areas of research you wear gloves. And anytime you're going to do PCR, you always wear gloves. And the reason for that is that this particular experiment is we're trying to amplify DNA found in plants. We don't want to amplify DNA found in our own skin or the bacteria that happen to be on our skin. We want to make sure what we're amplifying is coming straight from the source we're interested in. So we wear gloves. Now, this is probably more obvious to all of you than in previous semesters, but when you wear gloves, you have to remember that you still are not supposed to touch your face. You're not supposed to rub your eyes. You're not supposed to wipe your nose or do anything like that. That's just the same thing as not wearing gloves at that point. Make sure you read the entire protocol. I know you're not doing this procedure specifically, but make sure you read the whole protocol. Associated with the DNA extraction is worksheet 1.2. It's due Thursday. Make sure you complete that. The steps, just to go through them real quick here, just so you understand why we're doing some of these steps. Protocol in your lab manual goes through it pretty well, so make sure you look at that in answering your questions. Just a few words of wisdom as we move through here. Make sure you label your tubes. It seems simple, but I see it all the time where someone is working at the bench and they don't label their tubes, or they label them illegibly, and then when you go on to a later step, you don't really know what you're looking at, and then you have to restart everything. Steps 3 and 4, what protocol requires you to do would be to chop up around 75 to 100 milligrams of plant leaves, and the key to this is to chop them up really tiny. Mince them up really good because you want to make sure that it's easy for the DNA to be extracted from these tissues. After you've done that, you place it in a lysis buffer, and you take this metal rod and you squish it all together and break those cells up, and then you spin it in the centrifuge to separate out the liquid that contains the DNA and all that cell debris at the bottom. In the procedure, you put these in a new tube. Make sure those new tubes match the same labels as before. You add 400 microliters of the supernatant that came from this centrifuge step, and you add ethanol. Ethanol is important to add because what ethanol does is it precipitates the DNA. Without the ethanol, the DNA just stays in solution. You then take that solution, you add it to this column, put it in a microfuge, spin that tube. The supernatant that comes through, we get rid of that. Now, sometimes we put that in a tube and save it, particularly if it's really hard to get this source of tissue, just in case something happened and the DNA is actually here. for this situation here, we'll assume that the DNA attached to the column. Now, as you're thinking about this, why is it that the DNA attaches to the column? What property or what charge would that column have to have in order to bind the DNA? Because remember, the DNA has fallen out of solution because you put the ethanol in it. And so what's preventing it from going into the supernatant? Somehow or another, it's sticking to this column. And it has a lot to do with the charge. So think about that and why that might be so. We add wash to it after this step here, and that just makes sure that all of that extra debris and things that are not the DNA that we're interested in are removed. We then place this column in a brand new tube. We add water to it, make sure we add the water to the center of the column, and then we let it sit. This is a really important part. Often when students are working in my lab and they do a similar procedure and they don't get any DNA, I always ask them about this step here. If you don't let it sit, you don't give that water enough time to go through the column and solubilize the DNA. You spin it, and in this tube right here, you have DNA in solution here. Then the next thing you'll need to do is go to your Lab Exchange account and log in. And then what I want you to do is watch this video. I'm going to be honest with you. It's not the most exciting video to watch, but it does break down the process of PCR fairly well. And as soon as you watch it, it'll give me a signal that you've watched it, and then you'll get credit for it. I think it's about a 9-minute video. The second part of the lab was to do the PCR. Here's a link that you can go to, and it also talks about PCR and how it's very similar to DNA replication that you're learning about in lecture. Even though you're not specifically doing this in the wet lab, it's really important that we think about controls and why we use controls. So the first one here is the negative control. And what we're going to do is use water instead of the genomic DNA that we had in that tube after the DNA isolation. We're just going to use water, no DNA. So we would expect no amplification of the DNA because there should be no DNA in there. But if we do get a band in the gel next week on the negative control, that tells us that we had some sort of contamination somewhere. So it's an incredibly important control. If you don't do it, you really can't trust your results later on. Then we're going to have two positive controls. The first one is going to use a plasmid that contains that gene that we're sequencing. This should work amazingly well because we didn't have to isolate it from the tissue. So there's none of that extra background or steps that are involved that could have caused us to lose the DNA. This is essentially a tube that is bought from the manufacturer that has known DNA in it. So this is a great positive control. It will work if everything is set up right with the PCR. You will get a product when you run the gel in the next lab. So it's a good positive control. When you see a band here with the gel electrophoresis in our next lab, it tells you that the PCR worked. It might be that our experimental ones didn't work, but this positive control is important because it says everything was there. There's no reason it shouldn't have worked unless you made a mistake in isolating the DNA. The next positive control is using Arabidopsis genomic DNA. This is similar to the plasmid DNA, except the plasmid DNA is just that gene, whereas the Arabidopsis genomic DNA is all of the genomic DNA together. This should also work because you don't have to isolate the DNA, so there's none of the extra steps that could cause you to lose DNA. This is just a tube of DNA that is supplied to us by the manufacturer. So this is a good positive control. If it works, it tells us that the PCR should work. So that's the second positive control. Real quick, just so we're clear on the difference between these two positive controls, as I said, the plasmid here essentially is just this GAPC gene by itself. The process of PCR should have no problem amplifying this gene. Now the genomic DNA has all the DNA in Arabidopsis. It also has the gene that we want to sequence here. And so again, it should work because there are not any of those other debris or possible mistakes that could have occurred during the DNA isolation. So again, this should work out just fine. Okay, so this lab period, we're doing the initial PCR, which is going to isolate and amplify the gene. For the next lab, we're going to learn about something called nested PCR. And this is a way just to increase the specificity for the gene we're looking for. And we'll talk more about that in the manual for our next lab time. All right, that's all I want to say for this particular lab. I'll remind you of the things that are due Thursday that are linked to Lab 1.2. You have to watch the video, and you have these 2 worksheets to do, plus the 1 lab activity. You'll turn these all in at the same time with assignments for 1.1, 1.2, and 1.3. Read the following for Lab 1.3. If you have any questions at all, make sure you let myself know or your TA know. If not, we'll see you later. Bye.